Thank you very much for the introduction, Fritz. Sorry, I'm just getting over a cold, so I wish I could smell my leg tonight. Everyone else can smell it later, all right? But let's get into just talking about the evolution of Nysius as a group of seed bugs, and with that odd creature, the wēkiu bug. Of course, this work can never happen without everybody else. So if you've been to my talk before, the list of all of the assistants keeps getting longer and longer. These are assistants from Manoa here towards the end, as well as here at Hilo throughout the years. So we've had lots of assistants, especially for a talk this broad. Nysius genetics, as well as the wēkiu bug. I've been working on that since about 2005. Funding and support is largely by the Office of Maunakea Management. TMT did fund some of the research where we found some other Nysius species through surveys and management. College of Ag, and then PEPS back at Manoa was also a funder of the Nysius genetics work. And then, of course, Gordon Bennett, our colleague at UC Merced. Just acknowledges not just funding. We work with lots of partners. DOFA, NARS, essential for any environmental work here in Hawaii. Bishop Museum, they did so much of the baseline work for the wēkiu bug that we've really built on the shoulders of many different scientists at the university, Manoa, as well as Bishop Museum, and of course logistics for OMKM, they help a lot too. All right, basic insect introduction here. Back to school, everyone. There originally were only found, before people arrived, 12 orders of insects in Hawaii. Overall, throughout the world, is about 32, depending on your current taxonomy or which book you're reading in which year. So that's less than half of the available major groups of insects actually colonized here in Hawaii and then diversified. That's an amazing thing because we're out in the middle of the Pacific. Again, looking at of those groups of insects that evolved here, there's only about 250 to 400 distinct lineages, again, depending on what literature you're reading and which genetics you're reading this year. And those all diversified into about 10,000 native endemic Hawaiian taxa, as well as then some arachnids, some other arthropods. What we always have to deal with is about introduced species and how we work with those as taxonomists to figure out what's new, what's here originally, and what to do now. This number is now probably up to about 5,000, with about 9 to 20 new species each year. We will not be talking about all the rest of the Hawaiian endemic lineages of arthropods, from spiders. You see this little camouflaged orb weaver here. Really interesting spiders are here in Hawaii as well. Myrid plant bugs. Some of you have probably heard of Drosophila flies. This is a scaptomyzoid found in the wet forests. There's also our psyllids that attack ohia but use it as a host, make those little galls on the leaves. They're beautiful little things when you actually see them not in what looks like a cancerous growth on a leaf of an ohia, as well as other leafhoppers, and then some of my other favorites, the Nesocidinae kind of flightless leafhoppers, which we'll talk about a little bit later. There are so many lineages, all deserving of attention and study. A lot of this work we kind of put together in a large, basically found all the entomologists, wrote it all together, said, don't forget about insects. It's the same thing we've been saying for 20 years, well, basically every 20 years. Again, always look at the insects in an environment and see what they are and where they've come from. Insects comprise the bulk of endemic biodiversity. About 70% of all endemic animal life, complex multicellular life, are insects. What about Hawaiian insects? This is not to scale, that's my classic joke. This was my one attempt at Photoshop. I thought it was pretty successful. The wēkiu bug is not that big. Arthropods. What is an arthropod? It's a jointed appendage having creature with an exoskeleton. So that's crabs, harvestmen, centipedes, scorpions, millipedes, spiders. This group, almost 500 million years old. That is a long time for evolution of different creatures. Insects are almost that old, 450 million years. The bulk of insect diversity happened kind of at the same time as insects developed wings as a major group, and at the same time that angiosperm plants evolved. So you've got an insect that's eating plants, co-evolving, having chemical warfare with your host plant, becoming host-specific through time, and then dispersing and becoming isolated in different areas, those are all tied together with a huge amount of diversity that occurred throughout the world. But how old are the Hawaiian Islands? This is a review paper from Cali in Holland back in, I think, 2006. Ages of the Hawaiian Islands. Big island of Hawaii, 500,000 years old as it was formed. Not very old. Kauai, 5 million years. The Northwest Hawaiian Islands, as old as 29, 30 million years old. That's not as much time to evolve different lineages as the rest of the world has had, right? So how did insects arrive and diversify? Generally, along wind currents, especially for many of the small insect lineages that we find here, upper level wind currents dispersing up into the high atmosphere, getting into the jet stream, and then arriving somewhere. If it arrives, what does it do after that? Other ways that insects have gotten here, definitely through the water, through logs and soil, also seeds and birds. If they get here, what do they eat? If whatever creature or plant that they were consuming isn't found in Hawaii and they were very specific to eating one thing, what does it eat? Perhaps nothing and it dies. That's a filter for establishment and then diversity later. Here are some Nysius communis. This is on native Bidens. And then we have the speciation process. How does speciation occur within all of these groups? There's no hard, fast rule of how one insect can go through genetic isolation or host shifts and then diversify through time. Different lineages have done this in different ways. Some extremely diverse with hundreds and hundreds of species. And then there's groups that are a little bit more typical, like the Hawaiian Nysius, the relatives of the wēkiu bug, about 27 species. These are all different species, but they were from one original ancestor. I ruined the whole story for you. We'll talk about that. But they have diversified through time. And what's really interesting within this group is when they were originally described, Beardsley, Usinger, these scientists from the 40s, 50s, all decided that it was about probably 5 to 12 different introductions that arrived to Hawaii within this group, and then each one diversified to 2 or 3 different species because they're so diverse in size, shape, all of those characteristics. Here in Hawaii, the diversity of shapes, sizes, colors is more diverse than the rest of the entire world within this group of Nysius. Here are a large assortment of the Hawaiian Nysius, some of which are introduced here in red. But generally, they're small, brown bugs. It's hard to appreciate their beauty until you get them under a scope or see them up close or see what they're feeding on throughout the environment. And it takes a trained eye to see where they are. But when you're looking, you will almost always find a Nysius. So 106 species worldwide. So about a quarter of these occur here in Hawaii and are endemic to Hawaii and evolved here. There are other groups of insects. And when we talk about what insects eat, I'll go back a second. Most of these Hawaiian Nysius are plant feeders and seed feeders. They will consume phloem or xylem from the plant vessels, but generally they're also eating seeds. And seeds have a lot more nutrients, but it's a little bit harder to access when your only mouth part is a straw. So these bugs need to insert their straw, spit a bunch of enzymes in there, digest a seed, and then suck it back in. That's a precursor to the ability to eat different kinds of host plants because you've got a more wide complement of enzymes to break down food and consume it, which is really interesting for this group. There is one, Nysius wekiuicola and its sister taxon, Nysius that do eat other insects. But what is another insect? It kind of looks like a little seed. Outer coating, you got guts in the inside, like whatever nutrients are available in a seed. Maybe that's a reasonable thing if you have a nutrient complement to break down other food sources. How do we detect those host shifts? We don't really know yet. Heather's going to talk about it. Maybe. Some other Hawaiian lineages are very host specific. This is Drosophila molli, only consumes Prochardia palms here on this island. the larvae do, and probably only eating the yeast that is breaking down Prochardia leaves or fruits. What happens when something is very host specific and you get an introduced species that also wants to eat the same thing? Who's going to win? If you have no more Prochardia plants, what also goes extinct? Your other insect that also relies on that food source. But if you're more of a generalist, perhaps if one of your host plants isn't here anymore, perhaps it can still hold on. What if you're a generalist insect feeder and you're eating any dead insect that arrives near your face? That's what vacuum bugs do. But vacuum bugs aren't the only lineage in Hawaii that are carnivores where all of their relatives are probably not. This is our famous predaceous caterpillar, the Eupithecia, eating a Drosophila fly. That is not the endangered Drosophila fly I just showed you. It's actually a threatened species. This is just Drosophila melanogaster, one of the lab colony that we had a long time ago. But there are other groups that have shifted hosts in Hawaii as a very unique process that has occurred time and time again here locally. Insects are essential trophic mediators. They eat everything, everything eats them. They move energy and nutrients around throughout the ecosystem. That's an essential role they have. They're an essential food source for other insects, for other forms of life, bacteria, fungus, as well as birds. And they also pollinate other plants, which then grow, which then other insects may eat. Here's a few pictures of Nysius. How do you tell them apart? Nysius communis, we saw that earlier. Nysius terrestris, eating Chenopodium. Can you see it? There's your Nysius terrestris. They're very hard to find, but they do eat seeds of Chenopodium plants. It's quinoa, just local quinoa. Doesn't taste very good. Every one of these species have unique characteristics in size, shape, life history, how they grow, how they develop, and discovering that is really exciting and fun. In order to do that, you need to find them in ground and then identify them. What is the difference between Nysius terrestris and Nysius sinoculus? Can you tell? Look at this expanded wing angle. Of course you can. Oh, this is a little bit more flat. Genetically, they're nearly identical, at least in the genes that we've used already. Nysius rubescens, host plant, is it Dubaltia or is it Ohelo? We found them on both. Can it eat both? Is it only eating the seeds? This is actually a picture from one of my friends, Will Haynes, another famous entomologist. If you've ever heard of him, look him up. He was taking pictures of berries and had no idea that there were Nysius all over it. So he sent me these pictures because we were out in the field together, and I was like, oh, cool, thanks for getting those pictures of the Nysius rubescens. He had no idea he was taking pictures of them. Same here. See them all hidden? Let's zoom in. There's one. Here's one. We didn't have a reasonable host record for this species. It was just happenstance that we had some pictures of them now. And we could see actually feeding on the seeds inside the berry, not the fruit part, but actually having a long enough rostrum to pierce through the outer fruit membrane to get into those very small seeds of blueberries. Nysius beardsley. How do you tell them apart? This one's really dark, usually found on Dubaltia ciliata on Maunakea. The characteristic to differentiate this from other Nysius is it has hyaline stripes on the membrane part of the wing. Here's another one, same species. Are they the same? Hyaline stripes, white stripes. Color variation is extreme within Nysius specifically, and the variability of those characters is also extreme. It's really hard to identify these things. How do you identify them? Rostrum length. What fruit or seed might it be eating with a rostrum that long? How do you get functional characteristics out of a morphological character? Again, modeling color and patterns also used for taxonomically. Does it have only oppressed hairs or, my favorite, erect hairs? That's what's in the keys. But you have to get that under really good light to be able to tell the difference between very similar species. Again, expanded wing angle or not. These are all the characters morphologically that before genetics was used to define these species. And how do you learn all that? You go through the museum all day long. You've got your samples right over there, right? You ready to identify them? Yeah. What we found was lots of Nysius in different habitats throughout the Hawaiian Islands, looking through history, looking through the museum collections, looking through literature, and also field collections. There are wet forest Nysius, there are alpine and subalpine Nysius, and there are subalpine and dry coastal Nysius. If you think about it, a dry coast, hot, dry, wind-swept, is actually very similar to a high alpine environment for an insect. So, how do they arrive, and then how do they diversify? Do they arrive and stay, isolation, and then genetic drift? If something doesn't have wings, is it more likely to stay in one place? How is it going to lose its wings? How does it stay in one place? How do you have a loss of a character? How does it deal with other organisms? Does it find another organism to eat? Does something else eat it? The ability to change and adapt to those environmental and physiological conditions are really important for evolution. Here's me back when I was young. This is 2005 at Mount Ka'ala on Oahu. This is West Oahu. It's dry coastal scrub. Here's here on Big Island. This is Molokai, Kamako Preserve, as well as the summit of Mauna Kea. Nysius exists in all of these environments. Nysius, wēkiu bug, has lost their wings. But again, it's not the only lineage in Hawaii that is flightless with a trend towards losing their wings. The Nesosindis are, Micromis usingeri, a lace wing, which now has a beetle-like wing, which cannot fly, also an alpine endemic. Here's your progression of losing wings in Nysius. Long wings, long, large membrane, short membrane, almost beetle-like wings. This is the new species from Mount Ka'ala. Nysius hardii. This is the only membrane part of the wing. Look how small that is. And then there's the wēkiu bug. Extremely small wings overall, not just the membranous area. What is an alpine ecosystem? How does that influence evolution? Trade wind inversion above there, 9,000 feet or so, kind of cold, very dry, some glaciers every now and again. Those are the major perturbances in this environment, but otherwise it's pretty stable. Dry, windswept, occasional snow and rain. Here's the trade wind inversion, that's Mauna Loa. Subalpine environment is the transition zone to an alpine area. So if you have an insect that's surviving in a subalpine area and becoming isolated and adapted to those unique conditions, perhaps that's a precursor to getting higher up and surviving the alpine environment. The alpine environment doesn't have plants to eat. There's not a lot of things up there. There's some grasses, there's some other plants that occasionally exist, but one of the major nutrient inputs for the wēkiu bug, flightless insect, is eating other insects. They're scavengers. The wēkiu bug and the a'a bug on Mauna Loa are scavengers. They're micropterous, very small wings. They're not the only flightless Nysius, but they're certainly the smallest winged Nysius. Longest legs, longest head, just oddballs. They only occur about above 11,000 feet on Mauna Kea. This is their environment, rocks. When the air temperature is 0, cold, the rocks, this is the rock surface where the sun hits, gets up to 113 degrees Fahrenheit on any given day from just the solar input. And then at night, this is one of the coldest recordings that I had, negative 15 degrees Celsius. Extremely cold at night at the surface. But below the rocks, just underneath all of these, the temperatures are vastly modulated. It's cool, but not freezing. So they can get away from being frozen every night, even if the air temperature always freezes. Right here, right at the soil interface, above freezing. We described the life history of the insect. It hatches, from an egg, like all other Nysius, molts 4 times, becomes an adult. The process continues after mating. This is their food, aeolian debris, wind-swept other insects. This was a sample we did, threw down a meter quadrat, counted all the insects in there. This is their diet. We raise them in captivity. Vacuum bugs can only grow in warm temperatures, just like all other insects. They can only do that for about 8 hours during the day when it's sunny. Here's a fun picture from Maunakea about what is known about the overall abundance of vacuum bugs. Starting about 2007, more recently in about 2011, 2012, we've been more consistent with the same number of samples. The numbers of observed vacuum bugs fluctuates greatly per year. Different times of the year, different environments, but they persist. Here's a good picture. Good year, bad year. Bad year, and then the next year. High populations vary rapidly, which is what insects can do when the environment is good, when they can get their food, when they can grow. So, adapt or perish. Obviously, vacuum bugs can make it. They're up there. They've evolved there. They can thermoregulate against very major different conditions. Some of these lower elevation species that are dry coastal adapted species might have a chance surviving in a very similar habitat, especially desiccation-resistant things. What is a seed? Is it similar to a dead insect? We actually tested the gut contents of vacuum bugs that we caught in the field for plant DNA. They are feeding on the gut contents of other insects. And we're actually consuming the plant matter inside the stomachs of other insects. They also eat a lot of other parts of the insect, but they were positive for plant DNA. There are no other plants up there to really consume. So we're looking for evidence of isolation through population genetics of the vacuum bug. We collected a bunch of them. We used mitochondrial DNA for this study. These are all the original collection sites. All the green ones is where we collected vacuum bugs from throughout Maunakea. We looked in the orange areas, but didn't find them. This is through 2011. They've now since been found at Lake Waiau, but other areas not. Very limited genetic diversity. If you're into genetics, it's almost all the same thing. There's not a lot of genetic variation between each of those 9 differences. Sorry. What does it look like? At the summit, where we had the most samples, we have 9 different haplotypes, very limited genetic diversity. At these lower elevation habitats that have lower densities, almost all the bugs found were of the most common genetic type. That's a founding effect. This gene we're using is also only maternally inherited. It's only the females. But now we're looking at different species. Oh, crap. This is the exact same genetic haplotype on Mauna Loa for almost 2,000 base pairs of genetic data from the mitochondrial gene region. It's a lot of data. They have the same genetics, but they look different. They're described as different species. Morphologically are distinct, different genitalia. That's one of those great things that vacuum bug researchers and insect researchers in general use to describe species. So this is also indicative of one very large or multiple genetic bottleneck events and a population expansion thereafter. The time frame we have is not very useful for this kind of analysis. It's in the scale of thousands of years, tens of thousands of years. But this is consistent with our story. Center of genetic diversity, founding effects, and then breeding in these lower elevation areas, and occasionally maybe they die. But then you have another founding event of the most common genetic type. That could happen. But how did they get there originally? Remember, there were glaciers 14,000 years ago. Both Mauna Loa and Maunakea were glaciated. So where was the alpine environment? It was at a lower elevation. Perhaps the species of whatever Nysius you want to call it 10,000 years ago followed their habitat type that they've already adapted to and now became isolated. That's what would show that identical haplotype. Remember, they don't fly. The rest of the Nysius. Here in blue, this is the one lineage with all of the Hawaiian Nysius species. It's not very well resolved, except that they're all closely related to each other. Other Nysius here in Hawaii are introduced from Japan and are not native Hawaiian species. They still have not been undescribed. They need to be sunk. Some of these high elevation Nysius are related to other subalpine and mesic species. Beardsley, I showed you that before. They're high elevation taxa. What they're related to, man, they're hard to tell apart. They're probably all one major group that this genetic analysis didn't resolve. But what about the wēkiu bug? Its closest relatives are pretty well resolved. Nysius wekiuicola, Nysius blackburni from this island, but not, well, maybe Nysius blackburni from Maui. We just don't know. There was probably some predaceous creature that occasionally ate other insects on Maui that had wings that probably flew over here when this island got high and dry, just like Haleakala was or is. And Haleakala used to be a little taller, and then subsequently lost their wings. That's a reasonable way that could happen. We've also described a few new species. Marlena's in the audience here. We're still getting these officially described, but we're doing some illustrations because the pictures just don't do them justice. What do the closest relatives to the wēkiu bug look like? Small, mottled color, a little bit longer, mottled color, bigger hairs. If I was a betting man, I'd say Nysius wekiuicola is a little more closely related to the wēkiu bug, but we don't have evidence for that given the analysis we've already used. Maybe Heather will figure that out, right? Very similar, but with wings. There are some Hawaiian Nysius that are single island endemics, and all of the Hawaiian Nysius that are single island endemics are at the tops of their respective mountains on each island. Perhaps that's a pathway to speciation in Nysius. Go high up, get stuck, have low dispersal, and now you're a wet species. Originally, you were a dry, high species when the islands were dry and high. But islands erode, and now you've got single island endemics in wet mountains on each island that are almost all flightless too. That's really interesting. We don't really have that perfectly resolved, but that's my hypothesis. We're trying to get it a little bit better. The alpine environment is basically stable in a very long time frame. It's a predictable environment. Food is arriving out of the sky for you to eat. That's a pretty stable thing to have. There are many dry insect Nysius that can handle desiccation because of their hairs. They're basically hairy. They can keep their moisture in their body a little bit better and also protect themselves from solar radiation. A generalist scavenger probably evolved from something that wasn't too picky about what it was eating, something polyphagous, which most of those Hawaiian Nysius are fairly polyphagous. They'll eat many different things when given the option. And they don't have wings. So we already have evidence of a genetic bottleneck and barriers to migration as short as ½ a kilometer. We also have shared haplotypes across mountains and across species. What does that mean for your species concept? That's a tough one. We need to keep working on that. High elevation populations could become isolated. What are other mechanisms of selection? Time to hand it off to Heather. Maybe endosymbionts are part of that process. She's not there yet, we're not there yet, but we hope that we can have more information about how the wēkiu bug shifted its diet based on the rest of the microorganisms that help to digest nutrients and create other nutrients through biochemical pathways. look for our documentary that'll be on BBC next year. That'll be really cool. Lots of people to thank. I'll ruin it for you. This is my last talk here in Hilo. I'm going to a different university back where I'm from. I'm sorry, everybody. I'll come back and give more talks at some point. But I'm still involved with wēkiu bugs for here and forever. Heather, next one. I'll load you up.
Okay, thanks. Oh, right. I forgot. It's not just following.
So hold your questions. Hopefully we'll have time. I talk too much.
Thank you all for being here. Thank you, Jesse, for doing an amazing job, as always, telling us all about Hawaiian insects, and this time more specifically the Nysius. Usually, I think Jesse's talks are very broad, talking about many different types of Hawaiian insects. He's a very enthusiastic entomologist and has gotten a lot of people here on the Big Island and throughout Hawaii very excited and interested about insects. I am one of those people who was kind of hooked on entomology from taking one of Jesse's classes. And then I had the honor and privilege of working with him on my master's degree here. And now that I'm working on my PhD, I'm shifting just a little bit, even though my background is more entomology, ecology. For my PhD at the University of California Merced, I'm working with Dr. Gordon Bennett. And we're still working with insects, still Hawaiian insects, still Nysius, but we're looking at them through more of a microbial lens. And the reason we're doing that is because pretty much every single living thing on the planet relies on some sort of microbe or various microbes for survival. So we are going to look at the microbes associated with these insects to hopefully get information on how the insects have evolved, how they're able to survive, and yeah, use this microbial lens to kind of get a different sort of perspective on these insects here in Hawaii. So my presentation is broken down to 4 basic parts. First, I'm going to try and get you as excited about insects, get you as excited about microbes as you probably are about insects now, tell you why they're important, why I'm excited about them. Then I'll tell you what we already know about wēkiu bug microbes, which isn't too much. The project's still kind of new, but we do have some preliminary data. And I'll tell you a little bit about our study methods and some of our results and the future directions for this project. So first, why are microbes important? Well, in case you didn't know, we live in a microbial world. Hands down, they are everywhere. They're in us, they're on us, they're in the air, all over the place. This image is from the American Museum of Natural History's website. They have a nice section that kind of explains the human microbiome. So if you're interested to check that out. But it's also kind of how I see the world now that I'm learning about microbes. I look at people, I see microbes. I breathe the air, I'm thinking microbes, they're everywhere. And I guess before I started learning about them, I thought of them, perhaps how many of you might think of them, as bad guys. A lot of people think of microbes as the things that make you sick, things we're protecting ourselves from by using hand sanitizers, washing our hands all the time, antibiotics. If microbes are everywhere, all over us, then how come we're not sick all the time? Well, in reality, not all microbes are bad guys. A lot of microbes are playing beneficial roles for us and for every living thing on the planet. Some of the roles for humans specifically, they help our digestive system develop and function. They help us get nutrients and metabolize these nutrients from our diets. They even help regulate our hormones and our brains, so our moods and all kinds of brain function are partly regulated due to these microbes. And our immune system is in part regulated by good microbes that are protecting us from potentially harmful pathogens and things like that. So there's a lot of good microbes out there, but we are living in a microbial world, and we're kind of microbial beings. This is also from the American Museum of Natural History, and you can see here that 70 to 90% of the cells that make up our body are actually microbial. Only 30% are solely human. And even when it comes to our genes, which are the kind of the blueprints, the instructions for what our cells are doing, 99% are microbial. Microbes are doing all kinds of stuff that we don't think about and can't see. And even though a lot of us might think of microbes as bad guys, but Now, I told you that there's a lot of good guys. You can see here in the tree of life that microbial diversity is so much more vast than multicellular diversity that we picture on planet Earth. On the tree of life, humans are actually more closely related to corn than two types of bacteria are related to each other. So there's just so much microbial diversity in the world, and we're just starting to learn about what these microbes are doing. So another example, microbes are everywhere. This is from a paper that came out of some researchers out of UH and other universities, but you can see in this image that microbes are terrestrial in soil and rock habitats, freshwater, saltwater, free-living microbes. Some of them are symbionts, which means that they're living in and on all kinds of organisms, interacting plants, marine plants, fish, birds, everything. So they really are everywhere doing lots of things. But we're mostly interested for my research in their role in insects. And this is another image from a paper from 2014 that shows insects. This was a paper about aphids, so it's not our type of insect yet, but it kind of gives us an idea of how insects are relying on microbes. An aphid is feeding on plant sap, which doesn't really have a lot of nutrients, so they are probably relying on the microbes to synthesize amino acids and proteins that they need to survive. And this image just shows that some insects are getting their microbes either from their food source, which would be on the plant itself. Some are harboring these microbes inside of their guts. You can see here. Some harbor them associated with other organs, like their reproductive organs that you'd see up here. And some of the functions of these microbes are helping the insect digest plant polymers, which are the molecules that make up plants, helps to detoxify plants that might otherwise not be edible to insects. And lastly, they are, in many instances, synthesizing these nutrients for these insects. There was a study that came out of a research group in Japan that even showed when these insects that rely on microbes for nutrients lose their microbes, they do not develop very well. They have a shorter lifespan. So they tested this by applying antibiotics to the insects or increasing the temperature that the insects were reared in the lab to a point where the microbes couldn't survive. So either way, whether it's antibiotic or increased temperature, the insects lost their microbes and had negative effects on their development and life. So we thought, all right, these microbes are definitely playing important roles in insects. Maybe if we look at the wēkiu bugs and other Hawaiian Nysius microbes, we can also start to understand what role they're playing for these insects and how they help them to survive, and potentially what role they played in the evolution of all the insects that Jesse was talking about. So now we know general information about microbes associated with insects, but what do we know specifically about wēkiu bug microbes? Well, we don't know too much about the Hawaiian Nysius microbes yet, but we do know some information about Nysius in general and their endosymbionts thanks to a research group in Japan that looked at Nysius and their endosymbionts. This was in 2012. And they found that Nysius have a specific obligate endosymbiont called Candidatus Schneideria Nysicola. So it's an obligate symbiont because that means the bacteria only exists in the insect and can't survive outside of the insect, so you cannot culture it in a petri dish or something like that. And the insect can't survive without this symbiont. You can tell in the name Nysicola because it's a Nysius-associated bacteria. These Nysius also have a specialized organ. You can see it's marked with the yellow bee there. It's the really red part. On these insects, it's attached to the reproductive organs of males and females. So this organ is called a bacteriome, and its sole purpose is to house these endosymbiotic bacteria. Inside the bacteriomes are specialized cells called bacteriocytes, and those bacteriocytes are where the bacteria live. So this is what we know about Nysius and their endosymbionts, but we don't yet know if the Hawaiian Nysius have the same bacteria, what function these bacteria are playing. So that's kind of where we come in. And we did have some preliminary data. My advisor, Dr. Gordon Bennett, had a class at UH Manoa where the students actually did some research on several types of Hawaiian insects, looking at their microbes. And this is just the part relating to the Hawaiian Nysius. But the take-home message from this figure is that on the left-hand side, you see that's an herbivorous flying Nysius, Nysius terrestris. And on the right-hand side is the wēkiu bug. And you can see the left three bars in this chart have the obligate symbiont at the top. It's the turquoise with the star in there. So it does have that obligate symbiont. You can see in the wēkiu bug bars, the obligate symbiont is no longer there. So we know that the herbivorous Nysius elsewhere, and now we know in Hawaii, are relying on this microbe, but where did it go in the wēkiu bugs? We don't know yet. But what we do know now is the wēkiu bug apparently has lots of microbial diversity, way more than the herbivorous stuff. So we don't know what all those microbes are doing, or even the identity of all of them all the way yet. So this is kind of exciting because A, where did the symbiont go? And B, what's all that other microbial diversity doing in these bugs? So that's kind of where our project's going, is to try and figure out what's going on with these insects. So the way we are doing that is fieldwork, sampling. So the top are two images that I believe probably Jesse took, or maybe Marlena, someone in Jesse's lab. But the top is the wēkiu bug. I circled Maunakea and Mauna Loa because I don't have a picture of an a'a bug yet. So just bear with me that the wēkiu bug is representing both, because they both have similar morphological features and are doing the same kind of feeding behaviors and things like that. We're also broadly sampling lower elevation habitats to get some herbivorous relatives of the wēkiu bug and a'a bug to kind of get a baseline to make sure, you know, we get a few herbivorous flying Nysius and make sure we test what their obligate symbiont's doing, that it's there, and things like that. More specifically, when we sample on Mauna Kea, this is a picture of, there's Jesse there, Jessica Kirkpatrick from Mount Cam. My husband gets out in the field and helps me all the time, so it's kind of a team effort, but we get to go up there and look for the wēkiu bugs on the cinder cones. We move down in elevation just a little bit. That's where we found Nysius terrestris in the subalpine area. Then we move over to Mauna Loa. So on the road heading up to the observatory, where we still find ohelo, we search for Nysius blackburni. And then I've been up above there, but I haven't yet gotten any a'a bugs yet, but that's one of the goals for this summer, is to collect some a'a bugs to include in our study. And then we've also done some much lower sampling along Saddle Road on Kaumana Trail, and I've sampled in Hawaii Volcanoes National Park, but we didn't find any Nysius for this study in the park. But on Kaumana Trail, we got Nysius rubescens, which is one that Jesse mentioned, and blackburni also. So far we have terrestris, blackburni, and rubescens that we're going to use as our comparison for the two high alpine insects. Once I get all the sampling, we then take the insects into the lab, and then that's when we start the molecular work. So briefly, bear with me. This is kind of a quick schematic of how the lab work happens when we get these insects and try and figure out what's going on with the bacteria. We start with the whole insect, which in some of the images, Jesse had it next to a coin, so you can tell they're pretty small, maybe a little bit around the size of a grain of rice, or a little bit larger. So I get that under the microscope, dissect out the little bacterium that's attached to the reproductive organ. That bacterium is what I use for the DNA extraction. So you grind that up. DNA extraction is kind of like following some sort of baking recipe. So you break up the cells, you add different reagents, you incubate it, follow the steps, and in the end you get a tube full of DNA. Just looks like the clear fluid. And the DNA in the tube, if you can imagine what it looks like, is the double helix there, which is composed of strings of nucleotides that are these letters, the 4 letters that make up all life on Earth. So we share those with every other living thing. But in the DNA in the tube, we can't read those letters. So we send that off for sequencing. We're doing Illumina next generation sequencing, so we send that off to UC Berkeley. The machine reads in the genome. So the difference between genetics and genomics: genetics, you're looking at one gene. Genomics, we're trying to read the entire string of all the DNA that makes up an organism. So this machine takes in the entire genomic DNA, reads it in small chunks because it's not able to read the full string, spits back out these bits. So it read a little bit, reads it in little bits, gives us back the bits, and then we use computer programs, bioinformatics, to try and line those back up to get our consensus sequence back. So this is what we then work with to try and move on to this part where we actually find the genes. So when we got the genome sequence back, it was about almost 600,000 base pairs, so those individual letters. You put that information into, we use a computer program called Genius. So that does a lot of the hard work for us, and it reads the letters across and predicts where the genes are across the genome. It does that by finding these 3-letter sequences are called codons. So there's some 3-letter sequences that tell you to start reading, some that tell you to stop. And that's how it tells us, okay, this is probably where a gene exists. And this is an example of when you kind of zoom out. So this is one gene in, I don't know how many nucleotides are there, but it's definitely not 600,000. This, you zoom out, it's about 10,000 of them, and you can see there's several genes that were predicted across here. So you imagine you zoom way, way out and look at all 600,000 base pairs and all the genes. That's what we're working with. So we use software, this program called RAST, does a prediction of what all gene names are. I go in manually and I search each individual gene in the NCBI website, which is the National Center for Biotechnology Information. And then compare it against gene names in EcoPsych, which are the gene names associated with E. coli. So it's all this, many, many steps. First in the lab, in the field, then in the lab, then on the computers, and then we get the whole genome. And then we can start looking at what the genes are doing of the bacteria for this insect. So after doing all that, we have some preliminary results. This is the automatically generated output from RAST. So it's a prediction based on the genomic data we gave it as to what the symbiont's DNA or genes are doing for the insect. So I didn't create this list or this figure, but I did add a few, what do you call it, symbols here and a few labels just to kind of highlight the areas that are predicted to be major components to what the genome is doing. So you can see it's like amino acid synthesis, cellular respiration, protein metabolism. So a lot of different functions, but you can see there are many other options as to what the symbiont's genome or genes could be doing for the insect. The ones that I've whited out are things that were not predicted to be present. So one of them is like photosynthesis. So that's obvious, it's not a plant, but there are a lot of different things that could be going on here. And you can see over here, that's the length and the coding sequences. So that's the number of genes that were detected in our genome. So about 585 that we're looking at individually. So for my work, this is the predicted gene functions, but I'm going in manually and comparing our genes against the published literature to see if our symbiont is doing similar things than as compared to what was published with other insects and other symbionts. So this is just a small portion of what we're shooting for. So this is some nutritional gene pathways. What I found out, it looks like our symbiont, you can tell by the check marks, are indeed providing some kind of B vitamins and essential amino acids. It's likely that they are contributing these functions. The ones with X's, some of the genes are present, but the symbiont's probably not providing those to the insect. In strictly plant-feeding insects that are feeding on xylem and phloem, as opposed to seeds like these Nysius are feeding on, they're, I think, most likely relying more on the symbiont for these type of nutrients, but ours are feeding on seeds, so maybe they don't require as many nutrients from their symbiont. Instead, they could be relying on the symbiont for things like cell structure or maybe protection from pathogens or any of the other things I mentioned are possibilities. But this is just the start. This is how far I've gotten so far in analyzing each of the genes, but we're gonna keep looking and comparing and trying to paint the picture of what's exactly going on with these symbionts and these insects. So this summer, we still have to get back out in the field to collect more wekiu bugs. We want to try and get bugs from as many of the different cones where they've historically been found as possible, so we can look and see if they are sharing the same microbes across, you know, the geographic range and elevations. Definitely want to get some bugs to see if they have the same microbes. The story Jesse was saying, how we don't exactly know when they split, how they split, what the story is. It'd be interesting to know, do they have the same microbes? All that microbial diversity in the wēkiu bug, is it similarly present in the bug? So we're kind of curious to add the bug in there just to compare the microbes and see what's going on. And then we're, of course, including the herbivorous to know, once we know what the function is of the symbiont in the Herbivorous Nysius, we'll at least know what the wēkiu bug has potentially lost when they apparently lost the symbiont. We don't know if that specialized organ's still there, so we are going to, you know, compare the herbivorous with the non-herbivorous. And the main kind of goal is to figure out what these genes are doing so we know what role the microbes played, how they help the wēkiu bug survive, how they help the other insects to survive. And then we are working on a couple publications with this information, so the information will become available, so you guys can stay tuned and follow up and hopefully learn all about what we learn all about. And we're pretty excited to share that information. But thank you very much for your attention, for coming tonight. And it's a list of some of the people that helped us along the way. But yeah, I think that's about it. And Jesse and I will be glad to take questions if we have time.
Yeah, ask away.
Well, I have several questions, if you'll pardon me. Quickly, Heather, where you had the X's next to those genes, these genes are coming from sequences that you took from the samples that were coming off of the particular organ on their genitalia or something like that.
Right.
So if they're not coming from a symbiotic source, where do you suspect they're coming from?
Well, the X's were next to the either B vitamin or essential amino acid. So we're saying it's likely not contributed by the symbiont. There were gray genes. I guess I didn't explain the figure completely, but the gray genes were not present in the symbiont. The black ones were present in the symbiont. So all we're able to tell from that is which genes were contributed by the symbiont. The ones that were not contributed are likely contributed by the host.
Okay.
Yeah, but that is strictly the bacterial symbiont genome data that was shown there.
Okay. And then Jesse, I have two questions for you. Sure. One, why are you taking your genetic samples from the mitochondria? And two, you showed a graph with the populations of the insects in different years. I'm guessing those were estimated populations based on samples. What are the errors on those numbers?
Okay, yeah, so the two questions are, one, why are we using the mitochondrial genome for this analysis? Generally, the mitochondrial genome has been used in insects a lot for species and population level analyses. So for what we were really working on was just population genetics of the wēkiu bug. But since we had other Nysius available, because there's so much mitochondria in the body of an insect, because there are multiple replicates in every cell, we were doing non-destructive sampling, just pulling off like one leg so that we could still identify the insects. Just how that worked, we're much more likely to get results from a very limited genetic sample. So it's a practical reason of why we're using mitochondrial DNA for part of that. Generally, haplotype networks are used for defining how related groups of populations are in many insects. Nuclear DNA, we have some of that from the genetic analysis, but we just didn't have funds, or that wasn't really the ideal project that we were working on. So we had a small amount of nuclear DNA for the analysis. The other part of that is Nysius are poorly worked in general, so there's not a lot of reference genes to compare to. And even some of the nuclear genes we looked at, within the Heteroptera group, they're not mutating as quickly as other insects are. So within Hawaii, because there's only 5 million years of evolution, they're not very informative. So mitochondrial is, because it's a very robust region of the gene. So even when there are mutations, it still functions for cellular respiration. So this area of the mitochondria, the cytochrome oxidase 1 and 2, are just robust genes that mutate quickly enough is known about that throughout all of insects to have a good comparison. So that's part of the reason. And because it's maternally inherited generally, you just have a frame of reference for where your genes are coming from in that way. All right. The other question was the abundance of wēkiu bugs. That is raw abundance. So that's a very general trend through time. There is no error bars because that is the observed abundance of wēkiu bugs for, I believe, how many samples? 95? Is that the standard? 95?
I think we have about 60 that are in
60?
wēkiu bug habitat.
Okay. So yeah, so it's basically the Office of Maunakea Management. We do our annual summer sampling. And from about 2012 or so, or is that when it was?
2013.
2013. Because there's the same number of sample sites and they're in the same locations. At about 2013, that's more consistent to show a trend, even though there's not real statistics involved. It's the observed abundance for the same number of samples. Yeah. Okay, so it's all over the place. Realistically, the problem is wēkiu bugs sampling is attractant in cups. So if you're going out the same year and you have a cold day, they're not going to be walking around. So it's really tough to estimate densities based on that analysis. Jessica's done a lot more other work about estimating other kinds of densities and growth through other more rigorous methods.
Thank you.
Yep. Any other questions? 8:04. We went a little fast. Oh, we have one more.
So how would you, Heather, when you take all of the, DNA and you smash it all up and you try to sequence it and get that layout, how would you know how many different types of microbes you're working with?
I'm going to repeat the question because we were supposed to do that, and I don't think either of us remembered to do that. So the question is asking, how do we know which microbes we're dealing with or how many microbes we're dealing with? So for the Herbivorous Nysius, the one that I worked with most recently, by dissecting out the specialized organ that we know is housing specifically that bacteria, we try and minimize anything that's outside of that organ. So the goal is to target that specific bacteria because we know nothing else is in there except that bacteria.
Oh, okay.
Yeah, so the figure that was shown up here, there were two, it was Wolbachia and the Symbiont. That was, I think, a whole abdomen that was used, so potentially there are other bacteria in there. But for mine, we're targeting just that bacterium as much as possible. And then when we send this sample off for sequencing, you indicate that it is bacterial. So then they're targeting or using bacterial methods to make sure you're getting back bacterial data. We did get some insect data back with ours, but using the bioinformatics methods, we're able to separate out which was the bacterial data and which is insect data. Mm-hmm.
Maybe one more question. We have a little bit of a late start, so maybe we're exactly on time. 8:06.
Can you explain what a haplotype is, just for those of us who have taken a long time to go?
So it's basically, because it's only maternally inherited, it's a haploid, so it's haploid part of that on the maternal side. So the term haplotype is used specifically for mitochondria.
Okay. I was a little confused when you were talking about the number of lineages that are independent, like in Hawaii. What were you saying?
Yeah, so the question is how many other independent lineages are that came into Hawaii, and I did gloss over that quite substantially. That's a different question. The problem was those Nysius that were in that analysis were described as endemic Hawaiian Nysius in the original descriptions. When we looked at just the cytochrome oxidase data, that sequence is identical to Nysius plebeius from Japan. So it's things that are described as Nysius frigitensis from French British is not an endemic Hawaiian Nysius because it is identical to sequence from all throughout Asia through another Nysius. And there's quite a few other Nysius that is available on GenBank sequence that we use as part of the analysis is trying to find an outgroup for our Hawaiian Nysius. There's a lot of other Nysius diversity that we still don't know, that we could find a closest relative for where the Hawaiian Nysius came from, but we just really don't have good data to show that. So those two taxa that were on that phylogenetic analysis were two taxa that are misdescribed Hawaiian species that are actually Japanese species that probably arrived. All the first records are from World War II. They got to midway, basically Third War. So that's why those Northwest Hawaiian Island Nysius are most likely not endemic to the Northwest Hawaiian Islands. They got there as hitchhikers. That makes sense?
So you don't know them?
We know that the genetic sequence is 100% identical to Nysius caveus and Nysius.
But what about all the other species?
All the Hawaiian Nysius species are at least monophyletic within that Hawaiian Nysius group, given the outgroups that we did find. And we only have about 10 other Nysius that have any sequence of those 170 species. So from what we have, it's most likely supported as a monophyletic group for right now, but that could, of course, change if we get other outgroups. At least the closest relatives of the wēkiu bug are very, very similar, and we're really confident of the wēkiu bug's close.
So the Nysius looks so much genetically like the Nysius wekiuicola, but why is it so hard to find? I mean, is it just that there's no road to the top of Mauna Loa, and that's why it's hard to get there and hard to?
Honestly, I've never gone. So the Bishop Museum collected, all the samples that I did genetics. Yeah, that was multiple years of collections from multiple sites when Hawaii's and
And where did you find them? Did you find them eating the same? Were they also carnivores?
Yes. Yeah. So they had the same traps. It was during the same sampling years of 2005, 2006, 2007. So I had bugs from 3 different years from 3 different locations. And they all had the exact same haplotype, which was also shared with a subset of the wēkiu bugs.
So why are they considered separate species? If they have the
So
same
this
haplotype?
gene, cytochrome subunit 1, generally has a random mutation of 1 chance, and this is up throughout all insects, but Nysius are a little bit different. 1 random mutation of those 1,000 base pairs or so every million years or so. So what we have is completely consistent with a shared genetic type 20,000 years ago, because we wouldn't have seen a mutation yet. Because it's a slowly, it's just a little bit too slow to see that mutation within that gene for the time scale that we were trying to find information about. We also did some microsatellite work where we did see some differences, but there are only 4 informed sites, which is not published-wide. So we just don't have enough data to be conclusive. Except morphologically, they're pretty easy to tell apart. Bigger, different parameters, like it's pretty easy to tell them apart. And that's what we see with a lot of other Nysius. They're kind of easy to tell apart, but genetically, they're very, very similar. It's less than 0.5% difference, where usually in different species of insects, you see 5%. But that's why that gene is not used well for species limited designations for insects. It's just evidence towards it. It's not a definitive answer. All right. Long-winded, sorry.
One more question?
Yes, great.
One more question, or can I have 2?
We can always talk afterwards as well.
Okay, so I remember in one slide you showed a map of Maunakea, and in different regions geographically, you showed the variety of species, and I'm wondering first if I'm correct in assuming that the reason why you expect a greater diversity of species on the summit itself is because of isolation. And second, how you would know which of those species that you find are actually living there normally or are brought up there through wind?
Are you talking about haplotypes or species diversity? Because on the summit, it's just wekiu bugs.
It would be types. Sorry.
So is this a slide?
No.
I think he's talking about haplotype slide.
Yeah, you have different
Oh.
pie charts.
Okay.
That's the one. Yeah.
So this is just the 9 different genetic single nucleotide polymorphisms. So one nucleotide change within the mitochondrial DNA region that we looked at. This is relative size. So 52 samples of wekiu bugs only. These are only wekiu bugs. Half of those are all the same genetic type. So 25-ish for this. The next most common genetic type with one genetic difference is indicated in blue right here. So about 20 different individuals. Then there's a few smaller, basically we found haplotype changes occasionally in individual insects of those 52. Does that make sense?
Yes.
So sample size of 20 from Mauna Loa from 3 different years are all one genetic type. And it is shared with the second most common genetic type that's also found on Mauna Kea. So in these other regions, northeast and southeast, which wekiu bugs are much more less dense, they're harder to find. But if you just think of it in a raw statistics sense, you're more likely to get a founding mother in red, just statistically, to somewhere else. And because it's only the maternal line we're getting that information from, it would swamp out a signal, even if there were occasionally males that would interbreed with the females. So it's a kind of a raw statistical analysis.
But you know you don't have a bias because you have similar sample sizes.
What's that?
But you know that since your sample sizes are roughly the same, and they're, large enough that you don't have sample uncertainties.
Yeah, so there's an in-vivo analysis to check the probability that those populations are at least different from each other. And that's what that is showing, is they are statistically different populations, but they're just populations with different ratios of the total amount of genetics that's available or that we found. Okay? Generally in insects, like if you look at an entire population of, the most recent one that people have read is probably like the Kamehameha butterfly throughout all the islands. There's, I think it was like 46 different haplotypes. Big Island had maybe 15 or 20 different haplotypes on Big Island. The other islands had difference. So within insects, it's very common to see 40, 50, 100 different haplotypes within a species. It's very uncommon to only see 9. Like all of monk seals, actually the same gene region was used. I think there were 3. There's only 1,000 monk seals. So that level of genetic bottleneck is one of the things that this type of gene analysis can show. All right.
Thank you again.